Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're safe and well. It's a lovely sunny afternoon here in the UK, and I'm delighted today to be joined by my guest, Johannes Rauschnabel. I hope I got that right, who is the Director of Advanced Technology Development at Syntegon. And today we're going to be talking about a very topical subject, which is vaccine manufacturing processes and looking at it from the basic pharma business to COVID essentials. So, Johannes, thank you for joining me today. It's lovely to see you.
I'm very good. Hello.
Well, and where are you based? Where are you calling in from, Johannes?
I'm calling in from Kreisheim, which is in the southwest of Germany, and there we have our sterile pharma machinery manufacturing business.
Fantastic. Well, this is a subject very close to my heart today because, as Johannes will already know, I actually switched this interview because I have my vaccine, my first jab this afternoon. So this is a subject I really want to learn more about just because I'm going to be getting the injection this afternoon. So thank you for joining me and talking to me about this today, Johannes. But before we start talking about vaccines and how they're produced, perhaps you give people a quick overview of Syntegon and also your background as well.
Yeah, thank you, Rizwan. Syntegon is maybe more known under our former name, Bosch Packaging Technology. We were part of the Bosch Group and were sold in 2019 to the CVC Capital Group. And since January last year, we have the new work named Syntegon, which is artificial name, a little bit combination of synergies and TE for technology. That's my part. Go. So we want to be aggressive in the market and stay with our customer in constant contact. So being on. So that's a little bit of an explanation.
I didn't know that, so thank you for telling that.
Yeah, but we are one of the major packaging machinery manufacturers globally, also with a global setup of sites in Asia and US and Europe, and more than 6,000 employees. And by the way, we are one of the oldest packaging machinery companies. Our roots go back 150 years. So there's quite a bit of experience existing. And activities are in pharma and in food business as well. And pharma, that's our focus today in this interview. And my background, I'm a chemist from training, and I have a PhD in organic chemistry, but very honestly, that's many years ago. Since more than 25 years I'm in industry, and the last 20 years in pharma business, more or less. And the recent last 3 years, I'm heading what you already said, the advanced technology development at Syntegon.
Well, that must be a very interesting job, actually, looking at new technology.
Oh, yes.
So let's start talking about vaccines. And let's start with a very, very basic question. I'm sure a lot of people already know this, but for those who don't, and we get viewers who aren't scientists watching this as well, what is a vaccine and how does it work?
Yeah, that's a curious thing. If you go into history, vaccine is the name of a bacteria, of a virus, variole vaccine, which is the smallpox, the cowpox virus. And Edward Jenner then found out that people who had contact with this cowpox didn't develop the smallpox, which is a very severe and defacing disease. And so he started to think about vaccinating people to avoid that they get infected with smallpox. And this was very successful. And since then, vaccination is the word for whatever infectious disease immunization technology. And nowadays, it's more in vitro, but in the past, it was grown on livestock. And till now, it's also grown on eggs. So egg-based vaccines and flu and other applications are still very common.
Right, okay. And what are the common types of vaccines and how do they work?
So coming from Edward Jenner, we are coming from attenuated live vaccines. So he actually used serum from cowpox to inject it into people. So they got real stuff into it, but it was not really the real stuff. It was the cowpox, not the smallpox. But they have a very similar, how do you call it, cover of the virus chemically and from the morphology, so that the immune system learned with this cowpox vaccine that it was prepared there later on for the real stuff, for the smallpox. And so it all goes about the shell or the cover of these viruses, which makes the recognition for the immune system. And we've got two different immune systems in us. The nature of a human is perfect. So there's a fast one, which is very general, and this is the innate. So you have it from life start, and it will never change. It recognizes invaders and goes on them and eats them up. The macrophages are doing this job to gulp them, and then they're done. But there's another one which is adaptive and which can learn. And this is what we approach in the immunization. We learn this system to be prepared for later on contact with dangerous disease viruses. That's more or less in a nutshell explained how it works.
Right, but there's different types, aren't there? Because we talk about virus vector vaccines, toxoids, recombinants, and so on. So there are lots of different types, and obviously now we're talking about mRNA vaccines with the COVID vaccine. So there are a variety of different types of vaccines, aren't there?
There. You mentioned them already. Besides these attenuated, still live but attenuated vaccines, we have the fully inactivated one, which also comes from virus. But this one will be chemically inactivated, and then it also gets injected. But in this case, the immune system can learn, but the immune reaction is very slow and low level. So you have to add some adjuvants to make it faster, right. Which address different receptors and immune biochemical situations to make a faster and stronger immune response to it. And by this response, the immune system learns. And we have these toxoid vaccines, for example, tetanus is one of them. This is something, the tetanus virus itself is dangerous because it generates some chemicals inside in the metabolism, which are toxic. And in this case, you don't use virus units, but you use these chemicals, modify them, inactivate them, and train the system on these toxoid parts. And we have mentioned, and this is all over the newspapers right now, the mRNA, messenger RNA vaccines, which don't come from cells, which is more coming from DNA replication. I can go a little bit more in detail later on. And they give a wide variety of adaptation possibilities. So if there's a new variation, a new mutation, as we call it, of the vaccine, of the viruses, we can address them very fast with a new variation of mRNA. And it takes not this long cell growth, it can work more chemically.
Right, okay. So let's talk about the steps of the manufacturing process. What are the steps when you are sort of producing vaccines?
So due to the fact that the most of the vaccines are based on some kinds of viruses or virus parts, it always starts with a reactor, or in biology, biotechnology, they call it fermenter. So you put ferments into it and change a little bit the biological setting of the system. You have to feed nutrient media into this to get the cells all grow, and by that generate a high load of cells per milliliter. And as soon as you have a certain concentration, you should not exceed that, because otherwise you would poison the system. But as soon as you have a defined titer, they call it, titer, you stop that, and then you start to harvest, to separate, to clean, and there are different steps behind, which are quite complicated and time-consuming. But in the end, you come to the so-called drug substance or vaccine substance, the pure stuff, which then has to be blended with adjuvants, with preservative chemicals and so on, buffers, to get it to the final vaccine liquid.
Right. Okay. And what are the major differences then and challenges with mRNA, which obviously is in the news, like you said, over the virus vector vaccines?
That's a very good question, because it goes directly into why they are so slow with producing them. On the one side, they are much faster, and that's why we have the success story of developing a new vaccine with less than 1 year, which was in the past more than 8-9 years.
Yeah, sure.
So this is really a revolution, and this is due to the fact that you don't deal with cells. You start with a DNA strand with a plasmid, so-called plasmid, which is circular. You have to do some chemical treatment that you get it linear, and then you put ribonuclease acid polymerase to it, and then it starts to replicate, and from the DNA strands you read out the mRNA.
Right.
And this has to be cleaned, this has to be separated from all the DNA stuff. You have to fragment the DNA to get it rid of it, that you have only the raw mRNA, and then you get stepwise to the clean stuff, which finally has to be reduced for bioburden. There should be no microbe or anything in it, otherwise it would not be sterile. And then you blend it finally, and this is also a very important step. The mRNA is something injected into the body as pure stuff, which would be directly addressed from the innate immune system. The macrophages will cry out happily to get all this stuff to eat, but that's not what generates immunity. In this case, you have to shield the mRNA in so-called lipid nanoparticles.
Right. Okay.
And this is one of the last blending steps with the lipid nanoparticles. And then you can imagine they are little micelles, like soapy micelles, and in the middle is the mRNA. And this gets injected and finds its way into the cell, but not into the core. This is still shielded. It goes into the cell, and from there it goes to the ribosomes. And the ribosomes, they are the little production units of a cell, and they produce what was coded into the mRNA. And the mRNA has coded what was in the DNA strand at the beginning of the whole procedure. And you code into it the chemical structure of an outside of a severe virus. And so the body itself generates the antigen, which has to be addressed by the immune system. And then the immune system can train with its own antigen. So it's a self-contained, perfect technology.
It's quite amazing, actually, when you think about it, isn't it?
Really, but the tricky thing is about the carrier, the lipid nanoparticles. The tricky thing is about how to handle the mRNA, which has also some complex three-dimensional structure, not to change the structure. And the tricky thing is due to the structure, the mRNA is a little bit sensitive, and you have to do this all at very low temperatures. And especially in the latest step, the final fill finish, you have to keep it at you have to cold fill, and then you have to keep it at very low temperatures, not to avoid any decomposition.
So let's talk about the challenges around the fill finish then for COVID vaccines, because as you said, it is one of the most sensitive processes that you have to do and be very careful. So what are the main challenges, and how do you overcome those challenges?
Yeah, so one of the challenges is the fill process itself, because these new particles, they don't like, or these new mRNA vaccine formulation, they does not like any mechanical stress.
Right.
So you have to go with special fill systems. So like peristaltic fill or time pressure fill or rolling diaphragm fill. There are several fill systems which you can use, but others which you can't use, which are critical and would decompose and deactivate the mRNA.
Right.
You have to do this all at cold temperatures. And nowadays, we have the expectation of the regulatory body to go with very clean environments to shield the product, the vaccine, from the operator. So use barrier systems.
Yep.
And this is kind of challenge if you go to very cold temperatures, because then you have to avoid condensation on the barrier glass windows and all that stuff. And a second one is then after filling the product, it has to be closed properly, and then it has to go as fast as possible into very cold conditions. Otherwise, it will decompose. And also all the logistics have to be at very cold conditions. Depending a little bit on the formulation, there are vaccines out like Moderna, which accept storage at -20. Which is quite reasonable, but the BioNTech, Pfizer, this came with a different formulation, but was the first one. We are very happy about that. This needs -70 degrees Celsius. And this is from logistic a high effort to get this.
Right. But the AstraZeneca one then is not an mRNA vaccine.
No, that's an attenuated live risk vaccine, and that's the big advantage of those vaccines, that they are can be stored at refrigeration temperatures in household fridges.
So one question just comes to mind to me though, because you said obviously with live vaccines like the AstraZeneca one, historically they've taken much longer, whereas the mRNA ones, obviously because they've been able to be developed a lot quicker. So how has the AstraZeneca one been able to be developed as quickly then as the Pfizer BioNTech one?
Yeah, there was a not by accident, it was a lucky situation that they had already a basis of a virus vector which could easily be changed to go on the COVID instead of a different target. And I think they had used chimpanzee adenovirus basis, and they had it already developed, and it was in many applications and productions, it was approved. So they only had to change a little bit the virus microbiology. That's all. So and this helped them to be very very fast, even with this old-styled attenuated live vaccine approach, which is not old-styled at all, because most of the vaccines nowadays are this way.
Right. Now, well, thank you for clarifying that, because I'm sure there'll be people watching this thinking, well, actually, what's the difference between the two? And I know there's a lot of rumors go around about different types of vaccines and whether you should be using them or not. So thank you for that. So let's talk a little bit more about regulation as well, though, because I know that there is the GMP, new draft EU GMP Annex 1 looking to be approved very soon. So how will that have an impact on vaccine manufacturing?
So would be approved very soon. Your word in God's ears. I'm not so sure.
Well,
tell a little bit,
maybe they won't be there, but I don't
story. I tell a little bit the story why I'm not so convinced that it will be tomorrow, but hopefully the day after tomorrow. The Annex 1 is the major European and even worldwide guidance for good manufacturing practices for sterile medicinal goods, so veterinary as well as human. And the existing version is from 2009, February or March 2009. And at that time, we had only, I guess, 24 pages of this. And now there was in progress in technology as well as in practices, and we have new drugs. And so there was time to adapt it to the news level. And now they started to make a first new draft in 2017. It was published in December. I remember very well the day. And this one was more than 55 pages, so more than doubled. And it was a draft for consultation, and they got more than 6,000 comments for these 50 pages. So some of them were similar or the same, but they asked everybody to comment on this out of the industry. And then they needed another 2-3 years to come a new draft, which was published last year, February. And this was also for consultation, but only for a reduced group. They want not to be overwhelmed with comments. For industry associations, they selected, but the major ones were represented in this group. And they commented till end of July or in July. And now we are waiting for the new version since half a year. We know that it has to be harmonized with WHO and with PICS, which is a worldwide net harmonization organization for inspection practices. Right. And that means it takes time. And they again got hundreds of comments because they did not change so much to the first version. So it looks like they want to insist on something which is very much under discussion. The good thing about this is they opened for the first time in this field the risk-based approach. So quality risk management, if you have a different process, that's fine, as long as you can show that it works well and it makes safe and high quality products. Or if you have a critical point, then please come up with a critical control strategy. Very good, but they didn't do it consequently. They put too much details and examples into it, so the room for doing something differently is not there anymore. So right. There's a contradiction. I guess they will come up with something which is more lean and which we will love. But on technically wise, there are some challenges in that. The one is a positive one. We should use barrier systems to come to a higher quality. No contact of the operator to the product anymore. The operator is out of the room and he only can access through gloves, which is good. This is a positive one, but also some critical ones, which is, for example, the filtration. They want double filtration. They want a pre-use, post-sterilization integrity test, which is all very stressy for the product. And if you do the filter test with product, each filter contact with the product reduces because you get decomposition, you get stuck right. product and adhered product, and this reduces very much the output of your equipment, and which is not good for the vaccine situation we have right now. We want to get as much as possible in shortest time. So there are some things which have to be hopefully changed. And then we will have a new standard, which is globally a lighthouse, hopefully.
Right. Okay. Well, fingers crossed then for that. So final question, because we could talk for hours about this, because there's lots of questions I've got, but I don't want to take up your time now. But one final question. Obviously, historically, syringes have been the method to deliver vaccines to patients. Yep. But what are the other alternatives out there? Because there are people out there who really cannot deal with having injections and so on. So what are the alternatives out there, or that people might be able to see in the future for vaccine drug delivery?
We have different other approaches. A historical one is the oral vaccination, as we know it from polio 50 years ago. I remember getting this little piece, this cube of sugar, and then dip some drops on it, and I have to swallow it. That was my polio vaccination. But what I didn't know as a child at that time, it had some side effects. So some of the children got polio because this attenuated went back into a wild type, which was very critical. And so they don't do it in Europe anymore. There are some few oral vaccinations out, but not with polio in the world, but only a few. It's exotic kind of now.
Right.
What is more coming for flu is nasal. The flu
Right.
mist is a product which came up with swine flu. And this is a nasal application which works very well, but it has limits to the population ages. So you can't do the little babies below half a year, and you can't do it with people like me, 50 years and more. So
well, don't worry, I fall into that bracket as well, Johannes. Don't worry.
Yeah. And what is for those people who are a little bit more aged, let's call it that way, the golden and silver ages, this coming up is intradermal application, because the dermis, our skin, is the perfect address point for the immune system. We have these Langerhans cells in them, and if you get it into this little 0.3 millimeter deep dermis, you will have a very strong reaction of the immune system. So again, the human body is perfect. And the same, these injections can be done by syringes, but they don't go into the muscles. They just go a little bit into the skin. And another one is also addressing the dermis is microneedle array patches. We also worked in the past on these. And this is something to come up in the future. The organization in Seattle, path.org, is very strong in that and pushing it, because it has the promise of easy application. You don't need syringes, you just have a patch, this tiny little needles, and it needs not cold storage, cold logistics, and so easy to handle. But there are also some negatives coming with it. Did you really patch the full one? You can only put a little bit of your vaccine into it, and things like that. And we have needle-free injection, big promise in years, and very well and common in livestock vaccination right now, because they don't have to change needles, right. But it doesn't work with all kinds of skin, and the immune activation is also a little bit limited. But more or less, we will stay for much longer time with intramuscular injections. That's that's our future as it was in the past. But nasal will be, and intradermal will be something which will come more and more.
Fantastic. Well, look, Johannes, this has been a really fascinating conversation. Thank you very much for taking the time out to talk to me. If people would like to know more information, where can they get more information about vaccine manufacturing and production?
First, together with your blog, you will add some links, and I can very much recommend the homepage of vaccineseurope.com.eu, vaccineseurope.eu. That's an association of European vaccine manufacturers. They claim to produce more than 70% of all vaccine doses globally. And they have a very nice homepage with a special on COVID right now. Today I went through it, and there's a lot of information, different levels of detail. So it's really a pleasure to read that and learn from it.
Fantastic. Well, I understand also you've got some webinars coming up as well, haven't you, on vaccine manufacturing and getting involved. So I'll put the links for that above the video as well, so people can do that. So thank you very much for your time, Johannes. I hope you found that interesting. I certainly did. If you'd like to know more information, then you can check out the links which will be above the video, but also feel free to comment below the video if you've got any questions for Johannes, or message him directly as well. I'm sure he'll be happy to receive any questions you have. And yes, all this time he says thank you very, very much for the information. It's been a fascinating conversation. It's made me better prepared for my injection this afternoon. So now I'm feeling a bit more confident about having the injection. That's all good. And I just wish you, your loved ones, and everyone at Syntegon a fantastic 2021. And I hope you stay safe, healthy, and successful this year.
Thank you, Rizwan, and enjoy your injection.
Well, I will share a photo of my arm this afternoon on LinkedIn.
Okay, good.
with my passport, if I've got one, a little card to say I've had it done. Thank you very much for watching. Thank you, Johannes. As I said, if you've got any questions or would like to know more, please feel free to check out the links and get in touch with Johannes. And until next time, as always, stay well and stay safe. Bye-bye.
Goodbye.